All right, this is lecture 21, microbiology. We're going to talk about microbial infections, the respiratory system. The upper respiratory system includes all of these structures listed here, including the auditory canals and the ducts from sinuses and the nasolacrimal ducts, all of that, not just the mouth and the pharynx. And it's mostly shown here. It includes the auditory tube, or also called the eustachian tube, as I said, and also the nasolacrimal duct going from the nose to the eye. It is protected by nasal hairs, ciliated mucosal membranes in the nose and the upper throat, and normal microbiota that sometimes includes pathogens that are not pathogenic in this area of the body, but if they penetrate further or other areas, they could be pathogenic. But it certainly has plenty of normal microorganisms present. And that's the upper respiratory system. The nasolacrimal apparatus, which is part of that, is a series of structures on either side, ducts that lead from the eye into the into the nose. The tears that are produced by the lacrimal glands wash down across the eye and then down through this duct into the nose. Which, by the way, is why some respiratory infections could actually be picked up in the eye, and the person is infected because of this route of entry. The microbial diseases of the upper respiratory system are quite important. They actually encompass a lot of the infections that essentially everyone has during their life. It's a common site of infection and inflammatory disease. The different kinds of infections you can get, and the typical symptoms include inflammation of the following structures: pharyngitis, tonsillitis, laryngitis, sinusitis, and the most serious, epiglottitis. Pharyngitis can be caused by strep pyogenes and strep pneumonia, tonsillitis also, and some viruses. Should be viruses up here also. Laryngitis, again, pneumonia, strep pneumonia, strep pyogenes, and viruses. Sinusitis, often it's a bacterial infection. You get inflamed membrane and heavy mucus discharge that sometimes can completely plug the nasal passageways. Epiglottitis is inflammation of the epiglottis. It is potentially quite serious and a rapidly progressing disease, and usually caused by Haemophilus influenza. Upper respiratory bacterial diseases include streptococcal pharyngitis, which is commonly called strep throat. This is caused by Streptococcus pyogenes, group A beta hemolytic streptococcus, gas organisms. Also scarlet fever, same organism, strep pyogenes. These are serious infections. Another upper respiratory infection is disease, quite serious, is diphtheria, caused by Corynebacterium diphtheria. We are all vaccinated typically against diphtheria. And another fairly common infection is the infection of the middle ear, which, as you know, is connected to the pharynx by the auditory canal or eustachian tube. And the most common, listing them in the order of the most common cause of middle ear infections, or otitis media. Bacterial causes include Streptococcus pneumonia, Haemophilus influenza, Moraxella cataralis, Strep pyogenes, and Staphylococcus aureus. The least common cause. Okay, let's look at some of these. Strep throat or streptococcal pharyngitis caused by Streptococcus pyogenes or gas organisms is actually quite serious, particularly because it can progress to even more serious infections. It's the same exact organism that was responsible for some of the skin infections and soft tissue infections we talked about, and also cardiovascular and lymphatic infections we talked about. It will cause severe local inflammation and fever, often associated with tonsillitis and large lymph nodes in the neck and under the jaw. All the draining lymph nodes would be enlarged. You can see the tonsils in this picture. First of all, the back of the mouth cavity is oral cavity is really inflamed, and the tonsils on either side here are massively enlarged. So you get quite serious inflammatory effects. Otitis media is not an uncommon middle ear infection, not an uncommon complication if it spreads up the eustachian tube, auditory canal, up into the middle ear. That can happen particularly if you, if you're coughing heavily and blowing your nose very hard. You can get bacteria going up into the auditory canals and causing a middle ear infection. There are 80 different substrains defined by antibodies, so there are 80 different serological groups. These are strep throat or streptococcal pharyngitis caused by Streptococcus pyogenes is responsible for about 5 to 10% of pharyngitis, inflammation of the pharynx. The organism is fairly resistant to phagocytosis and produces enzymes that can help it progress through the tissues, including streptokinase, which is able to lyse blood clots, and streptolysins, which are cytotoxic. We talked about, I think, leukocidins before, which will lyse red cells. I'm pretty sure, white cells. I'm quite sure we talked about that. We also talked about maybe another enzyme, hyaluronidase. Anyhow, this organism can, oh no, that was for another organism. Anyhow, this organism can cause a serious inflammatory disease in the throat, which can get worse, particularly if you have further complications from it. One further complication is scarlet fever. It's caused by Streptococcus pyogenes, and you get a pharyngitis. If the organism can produce an erythrogenic toxin, the result is scarlet fever. The organisms that can produce this are lysogenized by a lysogenic life cycle type of bacteriophage, a virus that can infect bacteria, and they provide the gene coding for the erythrogenic toxin. The result being a red skin rash, very high fever, and a spotted strawberry tongue. This is what's called looks like the surface of a strawberry here on the tip, huh? It's more rare than it was. It was more common in the past. It's become more rare. The next organism that we're going to talk about is Corynebacterium diphtheria, which causes the disease diphtheria. It's associated with sore throat, fever, general fatigue, and not feeling well, malaise, swelling in the neck. And the main problem is you get this membrane composed of fibrin, dead tissue, and bacteria forming in the throat that can block the airways. The toxin associated with the organisms that can cause the disease, the toxin is called diphtheria toxin. It is present in some, not all, Corynebacterium diphtheria strains, because the ones that have it were lysogenized by a bacteriophage that brought the gene in, the gene coding for the diphtheria toxin. Non-toxigenic strains, the ones that don't have the gene, that can be present as completely normal microorganisms in the throat. You can find it in the throat of many people. Doesn't cause any problem because it doesn't have the gene coding for this toxin. We, as I said before, are all immunized against it. It's the D in DTP vaccine. T is for tetanus, P is for pertussis, whooping cough. So D is for the diphtheria. If a person does catch diphtheria, it is serious enough and advances fast enough that they are treated not only with antibiotics, but they also receive antitoxin antibodies to prevent the effect of the toxin. Quite a serious disease, actually. Okay, let's talk about middle ear infections. We mentioned before the organisms that are most commonly cause in descending order, most commonly cause these kind of infections. What you see here is a view inside the ear canal from the outside, and you see that there's a bulging out of the eardrum at the end of the canal. So behind the eardrum is the middle ear, and because of buildup of fluid and sometimes pus, you get pressure inside, which causes quite a bit of pain actually, and bulging out of the eardrum. So it's fairly straightforward for a person to look down the canal and see if there is an infection, evidence of infection by the sign of a bulging eardrum. These kind of infections are more frequent in early childhood. The frequency of occurrence in adults has been increasing over the past few years. Often it's treated with broad spectrum antibiotics, but in the end it seems that especially with cases of repeated middle ear infections in children, that antibiotics don't necessarily shorten the course of the disease process, and it doesn't, if a child actually gets many recurrent middle ear infections, what they do is they put in a very small drain into the eardrum, so the pressure is reduced with infection, and they eventually they'll remove the drain. Incidence of strep pneumonia and middle ear infections can be reduced by vaccine, pneumococcal pneumonia vaccine. Okay, those were bacterial infections in the upper respiratory tract. Now let's talk about viral infections. In terms of the common cold, most common colds are caused by rhinoviruses. There are many different strains of rhinoviruses, and about half of all common colds are caused by them. Some are caused by 4 different types of coronaviruses, and others there's no detectable cause, but it is seemingly some sort of viral infection. About 40% of cases, it's not known what causes it. Sneezing and congestion and increased nasal mucus secretions accompany these kind of infections, and actually help with spreading them. They're spread by typically inhalation of small droplets that people release. There are probably more than 200 different things that can cause this kind of infection. We've listed rhinoviruses, coronaviruses, but also there are some that aren't known. Rhinoviruses, as I said, there's more than 113 different types. It only apparently takes one viral particle of rhinovirus to cause an infection, as little as a single one. With most microorganisms, there is a minimal infective dose, higher than this. Rhinoviruses are quite good at attaching to adhesion molecules that are expressed on the surface of the nasal mucosa. Young children might get as many as 6 to 8 colds per year, whereas adults may get anywhere from 2 to 4 on average. Once you've had an infection caused by one type of rhinovirus, you're immune to it. So as you move through childhood into early adolescence and young adult and older adult, you get fewer and fewer colds because you've been exposed to more and more different types of viruses that can cause colds, and you have immunity to them. Which is why, sometimes kids in a family will all catch a cold, but none of the parents, or maybe just one of the parents, and occasionally everyone, both parents and all the kids, but it's rare. Fairly often the parents, adults, or one or both are immune because of previous exposure. Most effective protection are the IgA class antibodies that are found in secretions in the respiratory system. They're present in the mucus. Recovery normally occurs without any treatment, but sometimes a common cold can get a secondary infection with bacteria, and you get bacterial pneumonia on top of that. Okay, let's talk about microbial diseases of the lower respiratory system. Now, when we talk about the lower respiratory system, we're talking about from the epiglottis down. So that includes the trachea, the bronchi, bronchioles, and the alveoli. This part of the respiratory system is lined by a ciliated mucous membrane. It is a ciliated pseudostratified columnar epithelium with goblet cells producing mucin that turns into mucus when it contacts water. Until you get into the much smaller bronchial tubes, which do not produce mucus because they would get blocked. The diameter is too narrow. There are, in terms of protective mechanisms, besides that ciliated mucous membrane, which can catch and brush upwards dust particles and bacterial organisms, there are also alveolar macrophages within the space of the lungs that are able to phagocytose and kill microorganisms, fungal spores, bacteria, and even viruses. As I said, IgA is quite protective. The IgA in the mucus, saliva, and tears are these class of antibodies are the ones that are most protective if they're specific for the organisms. And the cilia are involved in the mucociliary escalator system, which constantly beats the cilia upwards and helps to clear and keep the trachea and the large bronchi clear of accumulated material and dust and bacteria. So here you see the respiratory tract. The lower respiratory tract is from here down, from the epiglottis. Here's the epiglottis raised up, but from the level of the epiglottis down, and the upper respiratory tract, so from here down, and the upper respiratory tract is from here upwards. All right. In the lower respiratory tract, inflammatory responses can be affected by or triggered by infection with bacteria, fungus, or viruses or fungi. They can all cause bronchitis, which is inflammation of the bronchi, bronchiolitis, which is inflammation of the bronchioles, the smaller the smaller conductive airways. And you end up with, in the lungs, you can have an inflammatory response, which is called, or infection, which is called pneumonia. This is inflammation that accompanies infection with a microorganism. So lung inflammation alone without infection is called pneumonitis. And infection in the lungs with inflammation is called pneumonia. All right. And this would involve the pulmonary alveoli. So in terms of bacterial infections in the lower respiratory tract, some of the ones that are pretty key, and these are names of the organisms, and these are the names of the diseases they cause. There's pertussis, or whooping cough, caused by Bordetella pertussis. TB, or tuberculosis, caused by Mycobacterium tuberculosis. And then different kinds of pneumonias, bacterial pneumonias, caused by several different diseases, several different bacterial organisms, and we'll talk about each of the ones that I've listed here on this page as we go along. Here's all of the organisms, actually. We'll talk about whooping cough and TB also. And we'll talk about these different bacteria that can cause different kinds of pneumonias. Streptococcus pneumonia causes pneumococcal pneumonia. Haemophilus influenza causes Haemophilus influenza pneumonia. Mycoplasma pneumonia causes what's called walking pneumonia, or mycoplasmal pneumonia. And Legionella pneumophila causes legionellosis, or Legionnaire's disease. So we'll start with pertussis, or whooping cough, caused by Bordetella pertussis. This is a organism that has the most virulent strains. There are different substrains. So the most virulent strains have a capsule. They're encapsulated. They will produce a toxin, pertussis toxin, which causes the systemic symptoms. They also have a tracheal cytotoxin in their cell wall that can damage or kill the ciliated cells in the ciliated pseudostratified columnar epithelium. What you get in the disease process is an accumulation of mucus in the lungs, and the infected individual goes through 2 stages with symptoms. There are 3 stages, 2 with symptoms. The first stage with symptoms is the catarrhal stage, just like a common cold, very similar to a common cold. The second stage is the paroxysmal stage. Here you have violent coughing, sieges. I don't know if sieges is the right word, but you have violent coughing episodes, I think would be a better word, that are accompanied by a whooping sound, because they're coughing so hard that they have to, and they're not breathing in well, so eventually they'll have to gasp for breath, pulling it in and causing a whooping sound as they gasp for breath between coughs during these violent coughing sessions. And then there's a stage 3, which is a recovery or convalescence stage. The disease is prevented by the DTP vaccine. It's the P in the DTP vaccine, and prevents the disease. And I've put in a link for you if you want more information about it. The photomicrograph shows some Bordetella pertussis bacteria associated with ciliated mucous membrane in the respiratory tract. Okay, let's move on to tuberculosis. TB, or tuberculosis, is caused by the microorganism Mycobacterium tuberculosis. It's a rod and it's acid fast, as you know. It does not stain with the gram stain. The lipid or wax in the coat makes it resistant to gram staining, but it is an acid fast type of staining organism. transmission is from human to human. There is another type found in birds, which can infect people who are severely immunosuppressed, people, for example, people in the late stages of HIV, late stages of AIDS. Treatment of tuberculosis is a very prolonged, drawn-out affair requiring multiple different antibiotics to treat. There is a vaccine. It is called BCG. BCG happens to stand for Bacille Calmette-Guerin. It was developed a long time ago. It is a live, avirulent form of the tuberculosis organism. It doesn't cause the disease, and they do use it as a vaccine, but it is not widely used at all in North America. It is used in parts of Europe and Japan, but has not been proven to be particularly effective, and that's why it's not used in North America. Okay. During a tubercular infection, what you get is the organism gets into the space of the lungs, and here you see it's inside the lung space, and will be phagocytosed by alveolar bacteria, alveolar macrophages, but unfortunately often is not killed. What you get are further accumulations of macrophages moving into the area where the organism is present. And they also get infected, and this kind of an accumulated mass of infected macrophages is called an early tubercle. And it develops further into a solidified area of macrophages, sometimes even with calcium deposits that have macrophages in them, and the bacteria are walled off inside this structure. So you have the bacteria, you get a central area that might be, I think it's called caseous, caseous. You get a central area that's dead cell material and bacteria, tubercular Mycobacterium, tuberculosis bacteria, and then a wall of macrophages that have the bacteria inside them, and then more macrophages on the outside, completely walling it off so that it doesn't spread through the body. But there are live bacteria. You can have live bacteria inside this. Now, unfortunately, if the disease progresses, what you get is that you get this tubercle actually rupturing into the bronchial space, and so the bacteria are in the center, in the caseous center, get released into the bronchial space and spread further through the lungs. And it becomes a progressive, chronic, slowly progressive, deadly disease. It used to be really quite terrible, and many people used to have it. Still in the world there are many who are infected with tuberculosis. Diagnosis is by the tuberculin skin test, in which some of the bacterial components, dead bacterial components, are put on a scratch in the skin. They scratch your skin, they put it on, and they look for what's called a wheel and flare. It's a DTH, delayed type hypersensitivity reaction. The positive test is an inflammatory response at the site where you had this needle scratch and the material put on your skin. That's a positive response, but it may be proof of infection, previous or current, or previous BCG vaccination, because if you did get a BCG vaccination, then you were immunized and you would respond to this kind of testing. Diagnosis also they'll do further diagnosis checking an X-ray or CT scan, and if they still suspect that you're infected, they may even do acid-fast staining of the sputum or trying to culture it. It's a very difficult organism to culture from sputum or from any sample, but I suppose some people do try to culture it. Typically, acid-fast staining would show if there are any organisms or not, and the X-ray. These are some statistics, a little bit old, I'll admit, but showing the drop-off, tremendous reduction in cases in the USA and North America. Cases in Canada, cases have also dropped, although the communities in the north have been more susceptible to higher TB rates. TB is still a serious disease affecting many people. It is one of the top 10 causes of death worldwide. It is treatable and curable disease, so it's quite unfortunate that it's still up there in terms of being widespread and a major cause of death in the world. Typically the drugs that are used are these, although I wouldn't ask you to learn that and memorize that, because treatment can change as new treatments are developed. It's worth knowing, however, that up to 2018 there were an estimated 10 million people contracting TB annually, and 1.5 million deaths in that year. About 10% are children, and about 250,000 children died in 2018 of TB. It is a leading cause of death in HIV positive or HIV infected individuals because they are immunosuppressed, and many people get exposed to TB but wall it off in a tubercle. So it doesn't progress, and they just have a calcified node that can be seen on an X-ray. But with severe immunosuppression, then it can emerge and come out and cause death in people who are severely immunosuppressed because of HIV. There are now some, despite the fact that there are powerful anti-TB drugs available, there are now multi-drug resistant forms of the organism that don't respond to those treatments. Nevertheless, new treatments, second-line anti-TB drugs have been developed, and so the disease is still, even if it is multi-drug resistant, is still treatable and curable. However, unfortunately, there have been there's been further development of drug resistance among TB organisms. These are called extensively drug resistant TB, XDR-TB, as opposed to MDR-TB. So XDR-TB is even more resistant. It is more serious, obviously, than MDR-TB because it cannot be treated with these second-line anti-TB drugs. About 6.2% of the MDR-TB cases when further investigated, turned out to be untreatable and are in fact drug-resistant TB, extensively drug-resistant TB. That's in 2018. So this is becoming more and more common and is really a serious development. Here are the estimated tuberculosis incidence rates in 2016. It is present in every part of the world, but in some parts much more prevalent. These are estimated rates per 100,000 population. There's no area of the world that is completely free of TB. And there are some areas that you have really a high incidence, greater than 300 per 100,000. It's very high. So still a very important disease to be aware of, and not something that you wouldn't see. In fact, I'm not sure right now, but in the northern areas of Canada, of North America, there are cases in these isolated communities, certainly, and they do come south for treatment, and you will get patients with who are positive for TB in the hospitals here. Okay, we're going to talk about bacterial pneumonia. I told you that there were several organisms that are responsible for most cases of pneumonia. I told you that pneumonia is a inflammatory disease of the lungs involving the alveoli and accompanying infection. We'll go through the different causes of pneumonia, the bacterial causes of pneumonia, and some of the characteristics of these different pneumonias. It is a major leading cause of death, particularly in elderly patients, but also in young individuals. The first one we'll talk about is pneumonia caused by Streptococcus pneumonia, also known as pneumococcal pneumonia. It's the most common cause of cases of pneumonia and is often called, as I said, pneumococcal pneumonia, but also typical pneumonia. So typical pneumonia refers to pneumococcal pneumonia caused by Streptococcus pneumonia. All the other pneumonias are called atypical pneumonia. So if a patient is typical pneumonia, has typical pneumonia, it's pneumococcal pneumonia. It's caused by Streptococcus pneumonia. And if it's caused by any of the other bacteria, it's atypical. It involves the bronchi and the alveoli with a high fever, difficulty breathing, and chest pain. The alveoli, in fact, fill up with fluid. You get edema. Not only edema fluid, edematous fluid building up in the lungs, but also a infiltrate of polymorphonuclear cells, mostly neutrophils, and also some red cells. There are many, many different strains of the Streptococcus pneumonia organism. The strains are defined by different antisera. They are identified because they'll react with different antibodies. So these strains are called serotypes. And the diagnosis is by culturing the bacteria out of the sputum sample. Typical drug that's used often, but I'm not teaching treatment, but often it's penicillin. Used to be a really high mortality long ago, before generally high mortality before antibiotics came in, but now most of the mortality is occurring in the very young or the elderly, but still a major cause of death in elderly patients. There is a pneumococcal pneumonia vaccine available, and it is a recommended vaccine for people, I believe, over 60 or 65. I'm not sure, but I think I gave that to you on a slide when we were talking about vaccinations. It is against these are the vaccines available that are against either 13 different strains or 23 different strains. Certainly something worth thinking about getting. Okay, the next most common cause of pneumonia is Haemophilus influenza pneumonia. It is often associated with predisposing factors. There are risk factors to developing it, including diabetes and cancer, poor nutrition. Alcoholics often will have this kind of pneumonia. There are antibiotics that can successfully treat it. It is one of the atypical pneumonias. Remember that typical pneumonia refers to pneumococcal pneumonia caused by Streptococcus pneumonia. Another atypical pneumonia, also called primary atypical or walking pneumonia. The reason it's called walking pneumonia is because the symptoms are a lower severity, less severe. and therefore the person often is functioning but not particularly feeling well at all. It is caused by Mycoplasma pneumonia, sometimes also called mycoplasmal pneumonia. So primary atypical or walking or mycoplasmal pneumonia. I try to give you these different names not so that you are confused, not to confuse you, but to inform you so that if you hear these different terms that you have some idea of that you have a record or knowledge that they are the same disease and it's caused by this particular organism. Rather than, only teaching you one and then you hearing other things that I did not teach you. All of these terms, mycoplasmal pneumonia, primary atypical pneumonia, or walking pneumonia, can refer to pneumonia caused by mycoplasma pneumonia. The unusual thing about this bacteria is it does not have a cell wall. And because it does not have a cell wall, only a cell membrane, when you look at it under an electron microscope at a very high magnification, it does not appear as coccus or rod-shaped bacillus, but you get this fried egg kind of appearance. The raised portion is not the nucleus, but the nucleoid area where you have the chromosome, and around it is the area of this just cell membrane and the interior fluid of the cell. So this is all cell membrane area, and this is the area where there's a chromosome. Remember, bacteria do not have any membrane-bound organelles, so it doesn't have a nucleus, but it does have a chromosome, and that will appear as a bump. So you get this what's called a fried egg appearance. This kind of pneumonia is more common in children and very young adults, quite difficult to culture. Diagnosis is typically using antibody identification or looking for the DNA that codes for it. PCR stands for polymerase chain reaction. I'm not going to go into the details of that procedure, but it's extremely sensitive if you're trying to detect the presence of any type of particular type of DNA. Okay, so that was streptococcal pneumonia and mycoplasmal pneumonia. And here you see mycoplasma. Let's move on. Here we have Legionellosis. Legionellosis is caused by the organism Legionella pneumophila. Not pneumophilia, pneumophila. It causes quite a severe pneumonia, high fever and cough. It is an organism that's found in natural waters that are stagnant and therefore can grow sometimes in air conditioning or humidifying units or systems, and they have to be kept clean. Some of these, if they're not operating on the basis of heat, forming a steam to humidify, then like these humidifying systems, if it's just cool, regular room temperature water, they can grow these organisms, and it's inhaled, and it's always by inhalation from some sort of area where they're growing, not from human to human transmission. But you get outbreaks of this on and off all the time in different places. The original outbreak in which it was identified as a human pathogen was in the 1970s, and it was quite a serious outbreak because of growth of the organism in an air conditioning unit on a large building where they were having conventions. There were a couple of conventions at the same time. And that's when people started realizing that this was a unique, separate type of disease caused by an infectious organism. Okay, those are the pneumonias and bacterial infections. Now we'll talk about viral infections in the lower respiratory tract that cause viral pneumonia. These include influenza and measles and chickenpox. If no bacterial cause can be determined for a pneumonia, if the pneumonia is not responsive to antibiotics, and there are no bacteria cultured from sputum samples, then a viral cause or viral etiology is the most likely suspected cause of the disease. More common in children than adults. In kids, in small children and infants, you can get a infection with what's called RSV, or respiratory syncytial virus. Respiratory syncytial virus has the unusual ability to cause cells to fuse. If you take a epithelial cell culture and put in a sample and it contains this virus, it will cause syncytium formation, fusion of the cells. Symptoms include coughing and wheezing that you can hear with a stethoscope, and diagnosis is by antibody serological testing for virus or the presence of antibodies against the virus. All right, let's move on to influenza that causes pneumonia in the lower respiratory tract. And we'll talk about influenza virus, which is a RNA containing a virus. The genome is RNA. Influenza virus, there are four types, but only 3 infect humans. So there are 3 types that infect humans, A, B, and C. And they are grouped into these 3 types on the basis of molecules present in the protein coat. I remind you that all viruses contain some kind of nucleic acid. In this case, it's RNA. It could be DNA and other types of viruses. And then it's surrounded, I'm going to make it in red, or orange. I'll make the nucleic acid in orange, and around it you have a protein coat. All viruses have a protein coat around them. Protein coat is made of capsids, remember? Say capsomer capsids, these proteins that form a geometric shell around the interior nucleic acid. And influenza is one of them, some bacterial or viruses have a cell membrane. The cell membrane is a phospholipid bilayer derived from the host cell, and the virus has it because as it buds out from an infected cell that it was produced in, it carries some of that cell membrane with it. Important thing to also remember is that viral proteins will be expressed on the surface of the envelope. In the infected cell, you get viral proteins expressed on the cell surface membrane, and as the newly produced viral proteins, as the newly produced virus particles in the infected cell are produced in the infected cell, they bud out and they take some of the membrane with them, so they have the viral proteins present on the membrane envelope that surrounds them. The 3 different types of influenza virus, type A, B, and C, are typed on the basis of the proteins within these proteins that are part of the protein coat surrounding the nucleic acid, which I've drawn in blue. Type A is the one that causes most epidemics and pandemics of flu. Type B causes moderate or local outbreaks only, and type C causes quite a mild disease. When you look at type A, which is the cause of most epidemics and pandemics, pandemics is a wider worldwide spread. Epidemic is a fairly sudden widespread, but not worldwide. When you look at the different type A influenza viruses, they are based on the surface proteins that are expressed on the envelope, the ones I've marked here in red and green. There are two of these proteins. One is called hemagglutinin, and the other H stands for hemagglutinin, and N stands for neuraminidase. These are virally expressed proteins that are expressed on the envelope that surrounds the viral particle. There are different subtypes determined using antibodies typically. There are different subtypes, so you can have, all kinds. There have been all kinds, H3N2, H1N1, H2N2, depending on which type they have. And you've probably heard about this kind of typing of the type A viruses. Whenever you hear this type of nomenclature, these are the substrains of type A influenza virus. Okay, so, and not all viruses are enveloped, but quite a few are, quite a few that cause disease in humans, particularly influenza viruses. I'll talk more about these proteins later. Here's a diagram showing the. I think my diagram is better personally, but here's the enveloped RNA viruses, type A, B, and C. Type A causing epidemics and pandemics. An important thing to realize is that there are animal reservoirs of type A flu, because quite a few animals are susceptible, including birds and pigs. So these can spread through wild bird populations and infect domestic farmed birds, geese, ducks, chickens, and also pigs can also catch type A influenza. As I said, the viral proteins are expressed on the envelope, and they are hemagglutinin and neuraminidase. These are the protein spikes that are embedded at the surface in the envelope. The hemagglutinin protein spikes are used by the virus to attach to host cells, and remember, viruses must attach in order to before they can penetrate into the host cell that they're infecting. So attachment is key, and that's mediated by the hemagglutinin spike. And the neuraminidase spike, this is a protein which has enzymatic activity and helps to release virus from a cell. Now, I'm not sure about how that mechanism works, but these are the kind of proteins that you find on the envelope. And different variations of the H and N spikes, these proteins, are what identify the different strains. And new strains develop all the time. These are inherently genetically unstable organisms that can undergo mutation and another process, recombination, and I'll explain that later. Type B causes milder flu, but can infect humans. It's only found in humans, not animals. And type C, the mildest form, does not cause any epidemics. Every year there's a different type of flu vaccine. I'll explain why. And typically it would contain a couple of different type A influenza viral particles. to immunize you against a couple of different types of type A influenza virus and sometimes one or two type B. Okay, here's quite a nice figure showing the interior RNA nucleic acid inside the protein coat, the capsomere coat that's made up of capsids, and then external to that is the envelope, which is phospholipid bilayer shown here all the way around here, and you see all of this is a phospholipid bilayer that's originally derived from the infected cell that this virus came out of, and in it, embedded in it, and on the surface of the envelope are the viral proteins, the N neuraminidase protein and the H for hemagglutinin protein. They're often called spikes because they appear in electron micrograph as spikes projecting outwards. Okay, let's I think it might be worth stopping here, and I'll pick up again on influenza at the beginning of lecture 21, part 2.